I'm Richard Lenski, and I'm a professor of microbiology at Michigan State University. And my research concerns the evolution of microorganisms, especially bacteria. And we've, over the years, done a lot of different experiments looking at the coevolution of bacteria and phage, evolution of antimicrobial resistance, things like that. But perhaps what the work that's become best known in recent years is this very long-term experiment that's gone on in my lab. So the LTEE stands for the Long-Term Evolution Experiment, and it's an experiment I started in 1988, and the idea is to watch the process of evolution in action as an ongoing process. We often think about evolution, dinosaurs or things that happened in the past. But with microorganisms, one can watch evolution as an ongoing process. And so the idea was to take a single strain of E. coli, start 12 populations from that single strain of E. coli, and propagate them day after day after day in the same environment, 12 little Erlenmeyer flasks, and watch how they change over time. When I started the experiment, genome sequencing didn't even exist. And we've now been able to use these new technologies. Wonderful students and collaborators have worked with me on this experiment, bringing new questions to the project. The bacteria have been doing really interesting things over time. Their mutation rates have been changing. They've been evolving new functions, and they've become more and more fit, better and better competitors over time. And because we can freeze these organisms and bring them back to life, I like to say we do time travel. We have a frozen fossil record of the bacteria from this experiment. And in addition to the bacteria having very fast generation times, the fact that we can bring them back to life out of the freezer has just made this such a fun project. I knew I wanted to run a long-term experiment, but at the time I started it, I imagined that a year would be a long-term experiment. So my original goal was to go 2,000 generations, which would take about a year of propagating the bacteria under the growth conditions that we give them. But you run it for a year, and then you have to measure some things at how the bacteria have changed. And then you have to write a paper. And then you have to submit the paper and get the reviews and revise it. And by the time our first paper was published, the experiment was already past, I think, 5,000 generations. And the experiment's just sort of taken on a life of its own. I called it the long-term experiment when it reached 2,000 generations, because that seemed so long. But now the experiment has passed 80,000 generations and has gone on for all these years. And it's gone on successfully, I think, because the bacteria do interesting things, the questions are interesting, and perhaps most importantly, wonderful students, postdocs, scientific collaborators have brought new ideas, new techniques to the experiment, and I like to call it the experiment that just keeps on giving. One of the interesting aspects of this experiment, one of the core questions that really drove me to do this experiment was, in some sense, the reproducibility of evolution itself. So by having 12 replicate populations, we can ask the question of, they're all in the same environment, they all started from the same ancestor, how do they look after a year, after a decade, after now several decades? How have they changed? And so in some sense, evolution is a mixture of reproducible and not so reproducible. It relies on mutations, which are random genetic events. But natural selection, as Darwin envisioned it, natural selection is sort of this driving force that if organisms live in the same environment, you might expect them to evolve in similar ways. So we're looking at this tension between the reproducibility and the idiosyncratic, non-reproducible aspects of the evolutionary process. And I think, again, it's part of what makes it a really fun experiment. But I don't think anybody else is going to necessarily go back and redo the entire experiment. Although, having said that, there are a number of groups that are also doing long-term experiments now with organisms like yeast and some other bacterial species. It's a tremendous honor, and I feel individually honored, but I'm also honored on behalf of the many dozens of people who have worked with me on this long-term evolution experiment and on other projects that we've done with evolving microorganisms. Science occasionally is an individual effort, but more often, and especially in the modern day, it's a team effort. I have been blessed to have large numbers, dozens of graduate students and postdocs, undergraduate researchers in my labs, colleagues from around the world who've collaborated on this and other projects. And so any award like this is a reflection of a wonderful team of scientists working towards the same aim, but sometimes diverging in their objectives. And that's, again, a big part of the fun of science. So it's a real honor and pleasure to be here. I've enjoyed the first day of the meeting, looking forward to more of it. And so I want to tell you about this experiment, which had very open-ended goals. And the next slide shows what sort of motivated me when I started this experiment in 1988. So one was thinking about the dynamics of adaptation. Would populations, would it look gradual? Would it be kind of a step-like process? And for how long could things go on adapting in a constant environment? Another question that has always intrigued me in both life of our individual lives and the history of life on Earth is sort of the repeatability of evolution. There's this deep tension between like the randomness of mutation as a process on the one hand and the Darwinian process of adaptation by natural selection. So if you start with replicate populations under identical conditions, will they evolve in the same way or will they diverge because of the randomness of mutations? And then thirdly is, could we integrate genomic and phenotypic data? And I have to admit, I had no real plan on how to do this when I started the experiment, but genome sequencing became a godsend in this experiment. So the experiment itself is really, really simple. It involves 12 flasks, 12 populations of E. coli that all started from the same strain, except for a mutation that causes 6 of the lines to produce white colonies and 6 to produce red colonies on a particular indicator agar. And that turns out to be important because it allows us to compete different genotypes. So they essentially can wave a little flag at us, whether they're on the red team or the white team. And every day, there have been some interruptions, and maybe we can have some discussion over refreshments later. There have been some interruptions in the experiment, but the basic principle is 365 days a year, we take 1% of a population and transfer it into 9.9 mils of fresh medium. The medium is a very simple, minimal medium with glucose as the limiting resource. There's a second carbon source in there, citrate, but E. coli, going back to its original definition of the species, is not supposed to be able to grow on citrate, at least not in the presence of oxygen. If you think about diluting these populations 100-fold and then regrowing them, they run out of resources after they've regrown 100-fold. So you go 2, 4, 8, in terms of doublings, it turns out it works out to about 6 2/3 doublings per day. That's how we count the generations in this experiment. And it's now past 80,000 generations since 1988. There's no horizontal gene transfer. Sorry for those who love the importance of horizontal gene transfer, but this experiment has no viable phages, there's no plasmid, and E. coli is not naturally transformable. But if you sort of do some very simple math of the number of cells and what we know about mutation rates, there have literally been in each population over the course of this time billions of mutations. Probably every point mutation has been tried many, many times over. Now, I started out my career as a graduate student actually studying the beetles. No, not the beetles from Liverpool, but I actually was a field ecologist. And somebody earlier today, in some very nice talks I heard, but somebody mentioned imposter syndrome. I'll admit, I've never had a course in microbiology in my life, but here I am. But I switched from insects to bacteria and microbes because of the rapid generations. But one of the incredible, powerful things for these kinds of experiments is that we have a frozen fossil record. The organisms can be stored frozen in a viable way and brought out of the freezer, and they're perfectly viable. And we can then do things like compete the evolved samples against the ancestor. So what do we get out of this? So one thing I'm showing here on the left-hand side, I'll put something on the right in a minute, but is what about the competitive ability of these bacteria? So this is showing just one of the 12 populations, and just for the first 2,000 generations. And relative fitness is basically the growth rate of an evolved strain relative to the ancestor, not measured like with a spectrophotometer, but based on head-to-head competitions in the environment where the evolution has taken place. And so you can see by the end of 2,000 generations, there had been about a 30% faster growth in this particular population, and it occurred by a series of steps. As mutations that start out very rare take a while to increase to the point that they're affecting the average performance in the population. This is now showing over 50,000 generations, not one population, but the grand mean of the 12 populations. And you can see that by 50,000 generations, the average population is growing something like 70% faster than the ancestor was, again, based on head-to-head competitions. Now, when I started this experiment, I was sort of imagining they'd reach an asymptote, that they'd hit some limit. You know, how long in this very simple environment could fitness continue to improve? And what I'm showing here are two mathematical models, both equally simple. They only have two free parameters. One of them is a rectangular hyperbola. That's the red line. That's basically saying there's an asymptote. The other one, the blue line, is something called a power law. And it decelerates like it's approaching an asymptote, but actually mathematically it has no asymptote. It goes on increasing, but the rate of improvement just gets slower and slower. So we had sort of noticed on some earlier data that whenever we thought about an asymptote, it seemed like the bacteria were smarter than us. They would get past what we originally thought might be an asymptote. So what I've done in this data set is I've truncated the data from this one data set and only showing using the first 5,000 generations. With the hyperbola, I asked, where is it going to level off? And it's that lower line. Well, they go past that. Give it 10,000 generations of data, and it says, oh, we'll move the line up. They go past that. Give it 20,000, it says, we'll move it up further, but they go past it. Whereas the power law not only fits the data, it predicts it. And we've actually taken this out to 60,000 generations, and this summer we're hoping to take it out to 80,000 generations. So this model is not only fits the data beautifully, it also is very predictive. Now you might say, and I've had people ask me this question, isn't that kind of crazy that at some point they're going to be just like doubling in 2 seconds or something that makes no biological sense? But that's not true. It's not true because the rate of improvement is continuously decelerating. So I sort of had this thought experiment. This was 50,000 generations. What if I give the experiment to somebody who does it for 50,000, and we do this for 50,000 scientific generations? That'd be about 1 million years, and it predicts that the doubling time would be about 23 minutes. And some of you will say, wait, isn't that the doubling time of E. coli? That is in a rich medium, but in a minimal medium, our strain started out with a doubling time of about 55 minutes. So, in fact, it doesn't make crazy value predictions, and in fact, you can extrapolate it out to the age of life on Earth, about 4 billion years, and it predicts maybe it'll get to like 10 or 11 minute doubling time, which is about where the fastest bacterial replications have ever been observed. Well, what about the repeatability? So this is showing, again, just one line that improved by about 70%. What if instead of competing things against the ancestor, we compete, say, red line number 1 against white line number 1, and we do that for a whole large number of points? What we see is that they're like most of the fitness trajectories are virtually identical to one another. There is slight statistical variation, but they're, on average, a typical pair of populations is only differing, even after 50,000 generations, by about 4% from one another, whereas relative to the ancestors, they've improved by about 70%. So very reproducible at the level of the fitness trajectories. So, started the experiment in 1988. In 1995, the first genome sequence was published of Haemophilus influenzae. Even into the early 2000s, I'd hear seminars by somebody who had sequenced this important pathogen or that important pathogen, and it all just seemed so expensive. But as all of you know, it just keeps getting cheaper and cheaper. So this is data from, or a phylogenetic tree from a paper with Olivier Tenéon in Paris, where we sequenced 264 complete genomes from this one experiment. So 12 populations, 11 time points, and 2 individuals, or 2 clonal isolates from each population. And I'm sort of deliberately showing it in a provocative way. You'll see, it looks like 6 of the populations aren't evolving and 6 of them are. What actually has happened is 6 of the 12 populations have evolved hypermutable strains, hypermutable phenotypes, where DNA repair and DNA metabolism have changed. We can rescale the branches and everything starts to look much more normal. Down in the lower left, I'm not sure this pointer really shows up, but at the lower left is the ancestor, and at the top of each of these trees for the 12 populations is the 50,000 generation clones, and a little below that is the 40,000 and so on, and early on, just above the flat line at the bottom are the 500 generation samples and so on. So each of these makes a very lovely little tree on its own, and there's been no cross-contamination during the course of the experiment. We had some putative cross-contamination at different points, and we would go back into the freezer and restart those lines. So what do we learn from sequencing? So one of the things that a molecular evolutionist often will do is look at the ratio of non-synonymous to synonymous mutations adjusted for the redundancy of the genetic code. And a value of 1 would be suggesting everything is just random genetic drift. A value below 1 would suggest most of it is purging of variation by natural selection. But what you can see is all of the values lie well above 1. This is an indication that most of what we're seeing, and by the way, this graph is only for those that are the non-mutators. The mutators, everything gets messier, but for the non-mutators, or before other populations became mutators, you have this very great excess of non-synonymous mutations, implying that these mutations reached high frequency because they are actual beneficial drivers as opposed to just passengers with only a handful of beneficial mutations. Well, what about the identity of the mutations? So it turns out we almost never see, there are a few exceptions, but very few where we see the exact same substitution at the nucleotide level in 2 or more populations. But we see this incredible parallelism at the level of genes and operons and functional groups. So E. coli has about 4,000 genes, just 57 genes that make up only about 2% of the protein coding genome have 50% of all the, little over 50% of all the mutations that we have seen of non-synonymous mutations in the non-hypermutable lineages. And we actually had found some of these genes even before whole genome sequencing. This is just showing 2 genes where 12 out of 12 populations, I think by 20,000 generations, each had a different mutation in those genes. What's interesting, well, another interesting feature is that among the genes with the strongest parallelism are ones with very core metabolic and regulatory functions. Pyruvate kinase, several genes involved in cell wall biosynthesis, DNA topology genes have changed in all of the populations. The stringent response has changed in about 2/3 of them. And some of thes genes are essential, like TOP-A and MRD-A. So what's going on that these high-level regulatory, often essential genes are changing? Well, people sometimes say, what's the selective pressure in the long-term lines? It's 37 degrees, it's glucose, E. coli loves glucose. The selective pressure, in a sense, is that we've removed much of the metabolic versatility that E. coli would have had as a natural organism. So I think what they're doing is fine-tuning a lot of these existing functions to deal with the extreme predictability that we have thrown at them in this experiment. There's a single resource, constant temperature, no phages or antibiotics, and so on. Now, I mentioned that the populations, some of them evolved hypermutability. This is showing one of those populations where for the first 20,000 generation it had accumulated, I don't know, something like 50 mutations, and then boom, it got a mutator. And what's interesting is 5 of the 6 populations that have become mutators have then subsequently picked up either compensatory mutations or actual reversions. And this is an interesting case where within this population, 2 lineages independently picked up compensatory mutations in the same gene. While we were, it takes a while to get those genome sequences, to do the bioinformatics, to publish the paper, and while I was giving seminars on some of it, Michael Desai at Harvard and Ben Good, a former student of his who's now at Harvard, said, don't just sequence those clones, just send us your whole freezer and we'll sequence everything. And that's more or less what they did. So they sequenced 1,440 metagenomes, essentially whole population samples at every 500 generation intervals through 60,000 generations. And they paid for it all. And I should mention they're sort of typically 50 to 100-fold X, so you don't really see things, it's not whole population, you're not getting the really rare mutations, but you're getting things that have risen to a high enough frequency that they start to look interesting. So, I had imagined when I started the experiment, I didn't imagine it would run for so long, but we'd see something like the top panel. What you're seeing on the y-axis is the frequency of alleles that rose to a detectable frequency. And if it rises from the bottom to the top, that's a selective sweep. And we see lots of selective sweeps in that top population. That's what I had imagined we would see. At the bottom, you see what happens with populations that evolved hypermutability. It just looks like a pile of spaghetti. It's hard to pick out the selective sweeps because there are just so many mutations. And in the middle, you see some of these very interesting dynamics where we actually sort of have a cross-feeding interaction where two lineages have coexisted in that population from about 30,000 generations out to 60,000 generations. And although this work with Michael Desai's group picked up more of those cases, we had actually known about some of them because we had seen colony morphological variation that seemed quite stable in some populations. And what we were able to show is it was an ecologically stable coexistence because we could mix different clones at a 1-to-1 ratio or a 10-to-1 ratio, and they would converge on an intermediate frequency, indicating that there was a mechanism for ecological coexistence even in this simple environment. So I give a partial summary before I shift gears. It looks as though, leaving aside the hypermutators, that most mutations that we observe that rose to a high enough frequency to be detected by these approaches are the beneficial drivers. Not all of them, of course. There's some hitchhikers, but E. coli wild type has a pretty low mutation rate. And we're also seeing a lot of parallel evolution. The fitness trajectories are very similar. The genomes, we're finding these targets that are very common across many of the populations. I don't have time to go into it, but the cell morphology has been changing in some reproducible ways. Many other things have been changing in a kind of a parallel way. So it's as though this is sort of what we call a fitness landscape or an adaptive landscape. We have this arbitrary ancestor, and they're climbing Mount Glucose. Some of them are, really genetic space is not three-dimensional. It's, it's extraordinarily huge, but it's as though some of them are climbing up the southeast face of Mount Everest, some climbing up the southwest face, but they seem to be doing things pretty similarly. That is, until around 30,000 generations. A postdoc went into the lab. One of the populations was much more cloudy than the other. It was growing on the citrate. I was convinced we had a contaminant that was using the citrate, but wisely, we began to check for markers. No, it's E. coli, it's a derivative of our ancestral strain, it's a derivative of the population from which it came, and yet a diagnostic feature of E. coli is that it's not supposed to be able to transport citrate under oxic conditions. By the way, our ancestor couldn't do it under anoxic conditions, and moreover, under oxic conditions, the traditional case of the rare cells that can grow on citrate under anoxic conditions, it requires co-metabolism of glucose. Ours can grow on citrate, oxic or anoxic, as a sole carbon source. So I had a fantastic molecularly trained postdoc, Christine Borland, who was trying to understand the genetics of this using P1 transduction. Couldn't really make sense of it. Suggested it might be an unusual mutation, not like a point mutation, but maybe some big inversion, so something that had not happened very often. Or maybe it was something that as they climbed up the southeast face of Mount Glucose, they sort of stumbled in this multidimensional space into the domain of attraction of being able to grow with one more mutation or a couple of more mutations on citrate. And to sort of presage things a little bit, both hypotheses are correct. It was a very unusual mutation, and it happened in context of the genetic background that was present in that population. So I'll say a little bit more about this. But anyhow, finding the genetics very frustrating. We took inspiration from paleontologist, historian of science, Stephen Jay Gould's book, Wonderful Life, where he talks about replaying evolution. So this is the Burgess Shale fossil deposit in northwest Canada, where about 500 million years ago, there were already diverse phyla of multicellular animals. It's one of the earliest fossil beds that recorded that. And he said, wouldn't it be cool, Gould said, wouldn't it be cool to replay evolution from that point and ask how repeatable things would be? And he calls it replaying life's tape. Shows you how fast cultural evolution occurs. Who's played a tape recently? Even vinyl's coming back, but I don't know anybody who's bringing tapes back. But he says, go back and start it all over and see if the repetition looks at all like the original. And he says the bad news, at the scale of planets, we can't do the experiment. But we can do it using the frozen fossil record of our viable bacteria. And the bacteria stay forever young in the freezer, but you can see those of us on the outside aren't staying young. So this is Zachary Blount. He's now a faculty colleague of mine while he was a graduate student who took on this project of replaying life's tape. He originally wanted to do it in flasks, but all he was getting was negative results. So he decided to, the basic idea was to take not just the population, but individual clones from the lineage that eventually became citrate positive. So start with the ancestor and 2,000 generation clones and 5,000 generation clones and 10,000 generation clones, grow them up, spin them down, and plate a gazillion of them on a minimal citrate agar, because at that point we knew our strain could grow even on citrate as a sole carbon source. So he saved Petri dishes in a cold room down the hall because he wanted to prove to his committee that he was doing hard work despite all the negative results. And so this was a year's worth of his Petri dishes. It's about a third of the Petri dishes that he used in this experiment over several years, 2 or 3 years. So he tested about 40 trillion cells in this experiment. Now, he couldn't run them all at once in an experimental design. He had to take, this clone and this clone and this clone, and he always included the ancestor. Well, with the replication of the experiment, about a quarter of his experimental effort, because he always included the ancestor, was the ancestor. And literally, out of 1 trillion cells, never saw the generation of a citrate-positive mutant. He left them for up to a month trying to see if he could get a colony on minimal citrate agar. So did he get angry? No. His experiment was a smashing success, and it was a smashing success that although the ancestor could never produce this phenotype, and no clone before 20,000 generations ever produced this phenotype, he did find 19 additional cases of citrate-positive mutants, all of which derived from a clone 20,000 generations or later, and most of them from around, I think, 27 to 29,000 generations, suggesting there was sort of a multi-step potentiation of this phenotype. So it's saying that the particular path mattered, that you had to accumulate some changes in the genetic background that made it even possible for this phenotype to be generated. So we started then applying genome sequencing to this one population even before we did some of the more systematic things. So this is a diagram of the ancestor in a phylogenetic tree of 30 genomes, and you can see where the citrate positive evolves in the upper right there, where it goes from yellow or orange to red. You might say, I'll bet this is one of those hypermutable lineages, and the answer is no. It's actually a lineage that became hypermutable after it evolved the ability to use citrate. But we thought about this in terms of like 3 evolutionary stages. Potentiation, what were the genetic changes that led to the ability to then actualize the phenotype by getting one more mutation? That's what I'm going to focus on in this talk. And then, of course, when anything emerges in evolution, it's likely to emerge in a sort of a not so refined way, and there's the opportunity to improve things, just like the earliest airplanes, or automobiles wouldn't be competitive with the modern forms. So it turned out the key mutation was a tandem duplication of a di- and tricarboxylic transporter. It's actually an antiporter. It's very interesting. It can bring citrate in and get rid of citrate, but it always exchanges di- or tricarboxylic acid, one for the other. And you might think, oh, a duplication. That second copy gave just enough enzyme or transporter to allow just enough growth that it could get over the hump. But what we were able to show, and I don't have time to present all of it, was it's really the second copy that's the key one here. Because in the course of that tandem duplication, there's new upstream elements in front of the second copy. And so the only expression that we can measure is coming from that second copy. So it's as though the cell has rewired an existing regulatory region with an existing structural gene in order to allow this transport to occur. And it turns out all 19 of those replays where Zach found something similar also involved rearrangements that seemed to be focused on the CYT gene. We didn't sort of prove the promoter hypothesis in each of those cases, but everything fit with it. Sometimes you can get a new promoter by deleting a region and bringing a new upstream element in front of the gene. So there were all kinds of weird events that produced this. So species are complicated. And And this, now I'm turning to some unpublished work that Zachary has done, Zachary Blount. So are they a new species? Well, like a lot of things in science, and perhaps especially in evolutionary biology, you got to think about definitions. So it's not just as though they're growing faster. It's a discrete novelty. It's something new, and it's a meaningful novelty. It's not like they've just become phage resistant. It's a meaningful novelty because it's a phenotype that is not associated with the history of one of the best studied species. I didn't talk about it, but you might have noticed on that phylogenetic tree that the citrate minus and citrate positive lineages actually coexisted with one another for quite a while. So the citrate positive lineage didn't drive the citrate minus guys extinct, at least not for at least 10,000 or so generations. So there's a stable coexistence, again, very much like the branching we expect to be associated with species. And while I'm speaking to a microbiological audience, I think some of you probably in your biology courses and evolutionary biology courses were taught this biological species concept that's often attributed to Ernst Mayr. It really goes back to Darwin at least. That the idea of species is not that they look different, it's that they're reproductively isolated from one another. Cats and dogs don't just look different, they can't make offspring together. Well, that presents a problem. Can we test this in our system? Well, bacteria are asexual, and yes, they have parasexual processes, but none are relevant to this experiment. But what if we introduced it? And we would love to do more of this, so if people know how to really combine genomes, I'd love to hear it in sort of an open-ended way, make hybrids that are 50-50 combinations of different lineages. But the idea is if the hybrids are competitively inferior in both the glucose and the citrate environments, that would be a barrier to gene flow. They might be able to exchange genes, but if the offspring of those recombinational events are less fit, then that's the beginning, that might call that incipient speciation. So can we test that? So the prediction is that as they become better and better adapted to citrate, is the valley between citrate and glucose getting deeper. And the cartoon version suggests yes, but can we empirically test that? So what Zach has done is he's generated or sampled clones from the earliest citrate out to about 10 additional thousand generations and competed them against the earliest citrate positive one to show that yes, they're refining. They're getting better and better, exactly as you would expect. What we can do by way of recombination is because this is a tandem duplication, they will recombine and get rid of one of those copies quite easily. So we can generate mutants that are citrate minus, but have different periods of time of evolving as a citrate positive organism. And the question is, are those lineages, or are those clones rather, becoming less fit over time if we pull the rug out from under them by denying them that one mutation that gave them access to growth on citrate? So they're confined to the ancestral niche, and yet they have this history of adaptation to citrate. So I'm showing the same graph on the left, and now on the right, we can see that in fact these hybrids are becoming inferior, at least along this genetic axis where we take away the key citrate utilization step. So now I'm shifting gears to something that was just published recently, again with Olivier Tennion and Alejandro Cus, where we brought to bear some of the new technologies that have emerged besides genome sequencing to look at the effects of large numbers of mutations in different clones. Now this is not the citrate. This is back to several of the non-citrate using lineages. So they used TN-seq to produce hundreds of thousands of mutants in clones from different time points. And then you don't do pairwise competitions like I talked about earlier. You do a bulk competition and see who makes it. So up here, all these, this is over just a few days, 30 generations, not 30,000 generations, just a 5-day competition, I guess. You can see many of the mutants. These are all insertion mutations, by the way, so it's not like fully representative of the long-term experiment. But you can see many of those knockout functions and they're deleterious. Others kind of in those gray tones are probably roughly neutral, but you see some that are beneficial mutations even though they're insertions. So in evolutionary biology, there's this concept of the distribution of fitness effects, the DFE. And so what I'm showing on the right are these estimated distribution of fitness effects. The two gray ones are the ancestors of the different colors, and the blue ones are some of the evolved lines. I think those are actually from 50,000 generations. And at first glance, it doesn't look like the distribution of fitness effects has much changed. But if you look just at the beneficial tail and put it on a logarithmic distribution so you get a finer look at it, at the lower left, you can see that there's a long tail of beneficial mutations that extends out to about a 10% advantage generated by insertions in the ancestor. But just by 2,000 generations, that tail has been eliminated. So essentially, that helps account for this diminishing returns that we saw in the fitness trajectories. The available beneficial mutations, the really big benefit ones, were accumulating early on. Then it's harder and harder to find those, and so we see this truncation of the beneficial effects. But another interesting thing, which I wasn't expecting, that they mined out of this very large experiment, was that the identity of what genes produce beneficial effects is also changing very rapidly. So on the left, what they're showing is the fitness, a selection coefficient, so 0 is sort of neutral. The black lines are showing that there's a whole bunch of genes that when they get a mutation give, say, a 3 to a 10% benefit. By 2,000 generations, those mutations, those same genes, if they get hit by an insertion, have become neutral or even deleterious, and another set of genes that was neutral or deleterious in the ancestor, the red lines, has become beneficial by 2,000 generations. And the same story then goes from 2,000 generations, where the red lines are showing beneficial mutations at 2,000 generations. Many of those insertion mutations would be neutral or deleterious at 15,000 generations, and another set of mutations has taken over for them. So that's suggesting that against this backdrop of sort of the statistical regularity of diminishing returns epistasis, where the benefits get smaller and smaller, that the identity of the genes is undergoing a lot of complicated changes, suggesting a lot of subtle epistatic interactions between genes. So I'm just showing one example where we are sort of able to track down the mechanism of this complex epistasis. So just looking at that red arrow, and don't worry so much about the heat map, but what it's doing, the arrow is pointing to two genes involved in cell wall biosynthesis that, as far as we could tell, could be very tolerant of insertion mutations in the ancestor. There was no measurable fitness effect with putting a transposon down into the middle of either of those two genes, and yet in more than half of the lines, by 50,000 generations, those same two genes had become essential. Whoa, from something that seemed dispensable to essential. And what had happened was some paralogs had been deleted that also were involved in peptidoglycan synthesis. So here's a very dramatic case of the identity of something that was neutral at the beginning becoming an essential gene that couldn't tolerate at least insertion mutations by the 50,000 generation mark of the experiment. So a lot of subtle genetics. And I'll just comment that the history of E. coli research has been very good at finding out sort of functions of genes, but a lot of it's based on sort of knockout genetics, whereas the subtle adaptations that we're seeing in these long-term lines, it's pretty hard to understand why some of these global regulators are having the effects, and cell wall biosynthetic genes and so on are having the fitness effects that they're having. I haven't talked about it, but we have also made isogenic constructs for a number of those genes and showed that the things that the repeatability or parallelism suggests are beneficial almost invariably when we make isogenic constructs. That's the correct inference. So parallelism is a wonderful signal, and in fact, some of the talks I heard earlier today were using that kind of logic. So what's next for this long-term experiment? I hope I have a few more good years as a scientist and writer. I'd like to maybe write some books, but I realize, okay, you know, I'm getting older, not younger. So if I want this experiment to go on, and by the way, I've asked a lot of people, should I, even as long as 20 years ago, I said, maybe we should shut down the experiment. People still, no, you can't. So I thought, I thought it was important to make plans. And so Jeffrey Barrick is a former postdoc, now a full professor, actually a named professor at the University of Texas, he's a biochemist by training, was key to bringing us into the genomic era when he was a postdoc in my lab. And so he's now running the experiment since 2022 for its second scientific generation. And I hope in perhaps 20 or 25 years, he'll pass it on to somebody for the third scientific generation, and let's see what we can learn between brilliant students, postdocs, collaborators with whom I've worked and now Jeff will work, and new ones, new technologies, and the inventiveness of E. coli and evolution as a process. So, I think, I know I'm standing between this talk and refreshments, so I hope I have explained these ideas well enough, and I'd be happy to talk to people over the refreshment break if there are questions, and I hope there are some, so come find me. I'll wet my whistle. Over 100 people have participated in this experiment, some very slightly, others extremely participatory. The work has been funded largely by the National Science Foundation and my university, Michigan State University. These are some of the many key players in the experiment, and I'd also like to end by thanking Jamie Hall and Caitlin Shaw for nominating me for this incredible honor, and for all of you for listening to me tell you about 37 years in 40 minutes.